In this lecture, we will take a close look at an individual who made the study of science and living organisms a lot more organized and really paved the way for um, scientists who came after him. So, in order to study living organisms and in order to keep a record of those living organisms, we use a couple of different sciences called taxonomy and classification. Classification kind of fits into taxonomy, and really what that is being able to classify and organize anything. Now, we're going to restrict that to talk about living organisms, but we like to have things organized and classified. If you were to walk into my closet, for example, you would see, my dresses hanging together, my pants hanging together, all of my shoes, together, black shoes together, brown shoes together. If you go into my kitchen and you look at my cabinets, you would see, all of the plates together, all of the bowls together, all of the pots and pans together. So, as humans, we like things to be classified, we like things to be organized. And in order to study living organisms, we need them to be organized as well. So, um, Carolus Linnaeus, or Carl von Linné, was his real name. He Latinized his name to Carolus Linnaeus um, after his study and after his work with taxonomy. But he was a botanist and zoologist, and he published Systema Naturae in 1735, which um, was a short book, and he proposed a system of taxonomy, what we refer to as Linnaean taxonomy. And what his idea was is that we would use the same type of naming conventions for living organisms, so that when scientists were talking about living organisms, or they were, you know, writing about living organisms, that they would use consistent terminology. And he revised this book and added to it over the years, and it became really the first really standard that we had for classification, for naming organisms. What he did is he knew a lot about plants, he knew a lot about animals, and so he divided the world into three kingdoms: plants, animals, and minerals. And of course, minerals were later abandoned, but that was everything at the time that he didn't know to be a plant or an animal. He used, within these animal and plant kingdoms, he used a hierarchical system of specific levels. And in his original um, book, he started with the kingdom. We had class, order, family, genus, and then species. Now we have put other groups in here since Linnaeus's time, but he really was the first who thought about how important it would be to classify living organisms and name living organisms. Now, scientists continued this work, and in the 1800s, there was a growing interest in the field of phylogenetics. And phylogenetics really takes another step into naming and classifying, in that we start to look at evolutionary relationships. And we can design what is called a phylogenetic tree. And in a phylogenetic tree, we organize the living things by how closely related they're thought to be along an evolutionary scale. In our first attempts at phylogenetic trees, the organisms were grouped based on their physical appearance. But now, of course, we have DNA, we have genetic and biological and embryological comparisons that we can use to really make sure that we are lining these organisms up or grouping them as closely as we can. So in 1866, Ernst Haeckel proposed another kingdom, the Protista kingdom, for unicellular organisms. So we had the plants, the animals, and the minerals. The minerals were abandoned, so we just had plants and animals. And so at that time, everything was classified as either a plant or an animal. And then Haeckel proposed this other group, and the Protista group became the third group. And then he later proposed a fourth kingdom, the Monarians, for organisms who didn't have a nucleus, so bacteria. So we had plants, animals, Monarians, and Protistas. And this was his rendering of the first phylogenetic tree from 1866, and he had his three kingdoms here, and then he added this fourth one a little bit later on. And then about 100 years later, in 1969, Robert Whittaker, who was an American ecologist, proposed adding the fungi kingdom. And he also included another level above the kingdom level, the empire or super kingdom level, and had basically this empire of the prokaryotes and the empire of the eukaryotes. And this five kingdom tree that we have here was really the standard for many, many years. Now we have modified this a little bit more. Instead of these super kingdoms or empires, we have domains. And then we have kind of restructured and looked at what types of organisms are included in each one of these groups. Now, we have DNA. We know all about DNA now, and we can use DNA to organize our phylogenetic tree. So in the late 20th century, we started to really organize these phylogenetic trees based on genetics. And so we get this kind of current phylogenetic tree of life that we have, developed by Weiss and Fox, and organized into three groups: bacteria, archaea, and eukarya. These became our domains. And so now we have three domains, and we still have that five kingdom system, but the kingdoms are divided up among the domains. Now, once we have living organisms organized, we have to figure out a way to name them. Our system of naming is called binomial nomenclature, which is a fancy way of saying two-name or two-word naming system. And we name organisms using their genus name and then the specific epithet. So humans are named Homo sapiens, and you'll see it written here. In binomial nomenclature, the genus part of the name is capitalized, the species part is lowercase, and then both names are italicized, or if you're writing them, they will be underlined. So it's very important as we go throughout microbiology and really all of science that you learn to write scientific names correctly. So capitalize the genus name, lowercase the species name, and then if you're typing, it should be in italics. If you're writing it, it should be underlined. Now, Taxonomic names were typically derived from Latin because Latin has really always been the language of science. But today, as we discover new organisms, it may be a combination of Latin or Greek or even some English-sized versions of those. Um, a lot of times when we're naming organisms, we take some type of specific trait and incorporate that into the name somehow. Um, for instance, um, Holoquadratum walsbii, um, the which is a type of archaea bacteria. Halo is derived from the Greek word for salt. So we know that this organism, this archaea, lives in a salty environment, and it has square cells, quadratum. You see there's Latin for four square, and then the species name, it's named after Walsby, who was the individual who discovered the bacterium. So we can name organisms really any way that we want to, but typically um, they're going to have a clue to some characteristic about the organism. Now, when it comes to studying bacteria, there is this definitive volume or multiple volumes on bacteria. Bergey's Manual of Determinative Bacteriology and Bergey's Manual of Systematic Bacteriology. They are the reference materials for classifying prokaryotes, and we use a little bit of that in how we approach and how we study the bacteria. Um, so many bacteria look identical. They're single celled, they don't have a nucleus, and so we have to have a way to classify bacteria that's based upon other factors, non-visual characteristics. So we look at their serological factors, we look at um, their metabolism, and we'll do some of these tests throughout the semester in the lab so that you can understand how certain organisms can be grouped together.